Type 1 hypersensitivity, and this hypersensitivity is IgE mediated. Before going into type 1 hypersensitivity, just let us understand what do we mean by hypersensitivity. So generally when a person is exposed to an antigen, then there is a localized inflammatory response to eliminate the foreign particulate. But sometimes what can happen is that the immune response can go berserk, and it can become so heightened that it leads to deleterious effects. So such a kind of inappropriate immune response is what is termed as hypersensitivity, and people generally connect with hypersensitivity more with the word allergy. So type 1 hypersensitivity is one of the types of hypersensitive immune reactions. There are type 2, type 3 and type 4 hypersensitive reactions as well. And in this session, we are going to look at only type 1 hypersensitivity. So just let us look at the learning outcomes of the session. So type 1 hypersensitivity is within the humoral branch. So you must understand that hypersensitivity can be both because of humoral system as well as cell mediated system, but type 1 is more associated with the humoral immune system. And it is a response to antigens, specifically termed as allergens. So in response to an allergen, the B cells can get activated and they can differentiate into plasma cells, which would begin secreting IgE. Now, this IgE can go and bind to the Fc receptors that are present on the surface of mast cells and basophils. These are granulocytes. And on binding of IgE to mast cells and basophils, they become sensitized. So, the sensitized cells on another stimulus can give rise to mediators that may give rise to a heightened inflammatory response, which in turn can also lead to anaphylaxis. So therefore, let us look at one by one the components of type 1 reaction. So the first thing that we will consider is allergens. Now allergens are the ones that are responsible for triggering the type 1 reaction. In order to understand allergens, let us look at how and why it is associated with IgE. So parasitic infections that are something that can happen can give rise to increased IgE levels. And so the IgE tends to facilitate clearing of the parasites from the system. What is also observed is that there are certain people that have a condition that is termed as atopy. Now, what does that mean? It means that such people have already a genetic predisposition towards a particular antigen. So they would respond to a particular antigen much faster than other people. Such people are associated with hypersensitive reactions, and these hypersensitive reactions are not to the parasitic infection but to several environmental antigens. Now, allergens are non-parasitic. Therefore, if you have to define allergens, then allergens are non-parasitic antigens. So we looked at these environmental antigens that stimulate IgE-mediated hypersensitivity. So it could be dust, it could be pollens, it could be food, say for example nuts, it could be say seafoods, or it could be a drug, it could be any of these that can lead to a hypersensitive condition. Now, atopic individuals are generally found to already have high levels of circulating IgE. And atopy has also been attributed to certain multigenic loci, especially those that have genes for various cytokines. So because they are already predisposed and they have certain cytokines getting, say, present in a higher amount, they are able to have a hypersensitive reaction. Now, the second component is the IgE, or what was originally termed as the reagent antibodies. Now, it was Prosnitz and Kastner in 1921 who demonstrated that when sensitized individuals are injected an allergen, there is a wheel and flare reaction. And because they were the scientists who kind of found this kind of reaction was called as a PK reaction. So what is wheal? Wheal is generally swelling. So it is edemated regions, and flare means a redness. So one can see immediately because of a certain allergen, either swelling and redness happening. And if this happens, then it is confirmed that it has or that there is an allergic reaction or hypersensitive reaction. But what is interesting to note is that they associated this with the serum antibody termed and they termed the serum antibody as reagents. Later, of course, these were identified as IgE. So when you look at the structure of the IgE, IgE has an additional, domain in the constant region, but it is always the C epsilon 3 region which can associate with the Fc receptors, while these are the antigen binding regions. So this is typically an IgE molecule. You can see how it has glycans associated, and these glycans in fact help in association with the receptor molecule. Now, the second constituent is, of course, the mast cells and the basophils. So, they have an important role to play in the type 1 reaction. So, IgE can actually bind to mast cells and basophils as they have Fc receptors specifically for IgE. So, the mast cells and the basophils both are granulocytes, and they have a high number of granules present within their structure. Basophils are granulocytes that are circulating in blood, while mast cells are majorly found in connective tissue, and that too in the periphery. Now, the granule content in the mast cells have histamine, heparin, serine proteases, and carboxypeptidases, while the ones in the basophil also contain histamine, they also contain tryptase, carboxypeptidase, and they also have chondroitin sulfate. So you can see that the constituents are more or less same. Histamine, we already know, is associated with vasodilation and constriction of the smooth muscles. So you know that this is already a primary mediator of inflammatory response. You have proteolytic enzymes which can also lead to tissue damage to a great extent. Now, so there are two receptors for the IgE that can be present on the mast cells, and that is, one is Fc epsilon R1 and the other one is Fc epsilon R2. Now, the Fc epsilon R1 is considered to be the high affinity receptor for IgE. And as you can see, Fc epsilon R1 has three polypeptide chains, that is alpha chain, beta chain, and the gamma chains. And the gamma chains and also the beta chain have a region which is called as ITAM. What is ITAM? ITAM basically is immunoreceptor tyrosine based active motifs. So it is at this point that the tyrosine can get phosphorylated. So phosphorylated tyrosines would lead to a conformational change and further a signal transduction. So therefore, when you have the IgE binding, the IgE binds to the alpha chain of the Fc epsilon R1 through its CH3 domain. So this is something that has been observed with Fc epsilon R1. Now, Fc epsilon R2 relative to Fc epsilon R1 is a low affinity IgE receptor, and its structure also is different. So you have only one transmembrane, one region that is spanning the membrane. So it is a single pass transmembrane receptor. And what is interesting to note is that there are 2 points at which there can be cleavage releasing this portion, and this portion is what is called as soluble CD23. You cannot observe the same with the Fc epsilon R1. So these are characteristics of the receptors that can bind to IgE, and these receptors can be found on mast cells and basophils. So now we look at how the IgE mediates the heightened inflammatory response. So what has been observed is that the cross linkage in terms of IgE is important. Now let us look at what do we mean by cross linkage. So let us consider that this is a mast cell and you have the Fc epsilon receptors for IgE, where the IgE has come and bound. When an allergen binds in such a way that it cross-links to IgE. So indirectly it is cross-linking to Fc receptors. You will find that the mast cell will get activated and this degranulates to release the mediators. What is also the studies therefore to confirm whether this is really the case, what was done is anti-isotope antibodies were used for IgE, which can come and bind to both the IgE, cross-link the two, and you will again have the mast cell degranulation. The same, so it's not just binding to the constant region of the Fc of the IgE that carries out cross linkage, but it is also if you have the anti-idiotype antibody binding to the antigen binding domain, you still have cross linkage and you can have the mast cell degranulating. So from this, what is very clear is that the cross linkage of the Fc epsilon receptor is what is important. So it is the receptor cross linkage that is of importance for degranulating the mast cells. So that was confirmed also by using a cross-linking chemical. So again, when the two IgEs are cross-linked with a chemical, you still have the receptor cross-linkage happening. If one uses an antibody directly for the Fc epsilon in such a way that the antibody binds to both the Fc epsilon, again there is a cross-linkage. And what is observed is that on cross linkage, if you have calciums being taken up by the mast cells, then you can have the signal transduction being mediated. So what was also important to note is that calcium plays an important role as a secondary messenger for taking the signal from the allergen into the mast cell. The cross-linkage of Fc receptors is needed to elicit a signal transduction, and the tyrosine kinases get active to phosphorylate the ITAMs of the Fc receptor, and thereby there is a signal transduction that leads to degranulation of the mast cells. Now the degranulation process itself, we'll just look at it in a little detail. So when you have the allergen binding to the two IgE molecules, which in turn are bound to two Fc receptors, as you can see over here, this is Fc receptor 1, this is also Fc receptor 1. So effectively the two IgEs are cross-linking the two Fc epsilon receptors, and when these two get cross-linked, the tyrosine kinases become active. They in turn are going to activate further effector molecules. So for example, they activate phospholipase C, which is converting PIP2 phosphatidylinositol phosphate into diacylglycerol, and IP3 goes to the endoplasmic reticulum. There it binds to rotenone receptors, and you have calcium being released into the cytoplasm. So intracellular calcium levels are going to go high. And plus, what also happens is that the diacylglycerol is going to activate protein kinase C. And this protein kinase C in turn plays a role in degranulation, where it helps in exocytosis of the granules. So the granules are migrating towards the membrane of the mast cell. And secondly, what also happens is that they are exocytosed out, releasing the granules. But you can very well note that due to the PTK getting active, you have the membrane becoming permeable to calcium, calcium channels, allowing the calcium to enter, and the endoplasmic reticulum releasing its store of calcium. So inside the cell, you have the calcium levels increasing. So the calcium is acting as a secondary messenger. Plus, what is also happening is that the phosphatidylcholine in fact is able to help the arachidonic acid get converted to leukotrienes and prostaglandin, and leukotrienes and prostaglandins are secondary mediators. They are also responsible, or they are pro-inflammatory in nature. So in all, what is understood is that secondary messengers are formed: calcium, cyclic AMP, DAG, etc. Now cyclic AMP is a transient level present, but this has also an important role to play in regulating the entire process. So mediators such as leukotrienes and prostaglandins, which are secondary mediators, are formed, and the cytoskeleton is aided in moving the granules towards the membrane, so that it can be released, and with the release, histamines, that is primary mediators, uh, are released into the um, environment. So that is something that is happening, and that is what is degranulation process. And so you can understand that the entire degranulation process happens because of an allergen coming and binding to two IgEs, which in turn are bound to two Fc epsilon receptors. So, what is very clearly observed about type 1 hypersensitivity is that uh, a person who is giving rise, or who is observed to have type 1 reaction, is actually previously exposed to the allergen. So, for example, in the first exposure, the allergen can be taken up by an antigen presenting cell, presented on its surface, and because of that, you have a Th2 subcell type getting activated, which in turn can activate a B cell, and the B cell further can interact with the allergen, and on this interaction it can differentiate into plasma cells and memory cells, and you have IgE being released. Now here what is important to note is that from IgM, what is formed in the B cells is IgE, and not even IgG, it is IgE that is formed. So class switching to IgE is an important development. Now, second time on, when the same person is re-exposed to the allergen, because you already have the IgE bound to the surface of the mast cells and the basophils, the allergen now goes and binds to the IgE that are already present on the surface of the mast cells and basophils, and there, due to cross-linking of the two IgEs, you have the degranulation happening. So very clearly, the sensitized mast cells are able to give a very fast response, and that to a very heightened response. So it does not take all this while to reach the stage. Immediately as the allergen comes in, you can have the response happening because the mast cells are already sensitized, the basophils are already sensitized. So we can say that it is an acquired reaction. So due to the first exposure, the person already has the antibodies. So since it is antibody mediated, we call it a humoral immunity. So it is associated with the fact that Ig is already existing and present on the surface of the mast cells, that you can have allergen going and binding to the mast cells directly, giving rise to all the mediators that give rise to an inflammatory response. So along with histamines and leukotrienes, cytokines too are released, and these cytokines in fact recruit neutrophils and eosinophils. So you have migration of neutrophils and eosinophils happening that would further enhance the hypersensitive reaction. So there are two stages of type 1 hypersensitivity. One, it can be an immediate effect. So immediate effect is within 2 to 4 hours itself, and the initial response to the exposure of allergen is therefore sudden. And it is mediated mostly by mast cells. So you can have constriction of smooth muscles because of bronchial constriction. So you can see that asthmatic people wheeze, have a wheezing effect. They have difficulty breathing. Plus, constriction of intestine can lead to cramps and diarrhea, or there could be vasodilation and so swelling or fluid, and also it activates enzymes, so there can be tissue breakdown. So all these can happen immediately due to the type 1 reaction. The second stage is that it can be late phase, and the response can be only after 4 to 12 hours after exposure, and this response can last for about 24 to 72 hours, and these are generally mediated by eosinophils and neutrophils. So let us take the example of asthma for example. So a person who is asthmatic, okay, the early response is that the allergen can bind to the mast cells which already have the IgE present, and this mast cell degranulates to release the primary and the secondary mediators, which can cause vasodilation, bronchoconstriction, and likewise. But when you have a late response, which is because of an antigen presenting cell taking up the allergen, presenting it on the surface, activating the Th2 subset of Th cells, and that releasing IL-4, which in turn will produce IgE. That IgE will go and bind to the mast cell, and the mast cell will then secrete a lot of mediators like platelet aggregating factor, IL-4, You have TNF alpha, that is tumor necrosis factor alpha, leukotrienes. All these are basically going to recruit eosinophils and neutrophils. And as you can see over here, when you have infiltration of neutrophils and eosinophils happening, then you can see how the basement membrane is definitely getting swollen compared to when you have the early onset response. So at both the early phase and the late phase response, you have several mediators that are doing the that is giving rise to a particular response. In case of the late response, leukocyte migration and leukocyte activation is something that very clearly happens. Now, you can therefore have a systemized anaphylaxis. What does it mean? A systemic, in fact, anaphylaxis. It means that you can have these mediators traveling through the blood or through circulation to a more broader area. While you have localized anaphylaxis, which is confined to a specific target tissue. So that is the difference between systemized and localized. This becomes more generalized. And so, for example, you can see rashes all over the body, then that is systemized. But if you have a rash only near, say, the place where the mosquito bit, then that is considered to be localized anaphylaxis. So systemic vasodilation and smooth muscle contraction can lead to severe bronchial constriction, edema, and shock. And it can be fatal. The fatality is because of bronchial constriction, where breathing becomes essentially a problem. It can be caused by bee stings, some foods, drugs such as penicillin, insulin, etc. Now, localized anaphylaxis is generally considered to be an atopic condition, and the target organ responds to the direct contact with the allergen. So, for example, somebody has eaten some food, then the digestive tract is the contact organ, and therefore it results in vomiting, cramping, diarrhea. So, skin is the direct contact tissue, then it usually gets reddened, inflamed, resulting in itching, etc. If the airway is the target organ, then what can happen is it can lead to sneezing or excessive running nose, wheezing, and asthma. So this is localized anaphylaxis. Okay, so type 1 hypersensitive can be both systemic anaphylaxis as well as localized anaphylaxis. What is the treatment? Generally, the treatment is two types. Mainly, it is pharmacotherapy, so drugs are given. So, the most common drugs are non-steroidal anti-inflammatories that are given. Antihistamines are given. So, for example, cetirizine is given, levocetirizine is given. So, what it does is it is going to counteract the function of histamine, and therefore you won't have a heightened immune response, inflammatory response for that matter. Steroids can be taken, theophylline or epinephrine can be given. In fact, for most severe hypersensitive conditions, epinephrine is given because epinephrine works exactly the opposite to what histamine does. So when histamine leads to vasodilation and bronchial constriction, epinephrine relaxes the smooth muscles and it makes the vascular system less permeable. So therefore, exactly opposite to what the histamines do. Plus, what it also does it prolongs or increases the cyclic AMP levels in the mast cells, and this because when cyclic AMP levels are high, then it can inhibit degranulation of the mast cells. So that is another important thing. The second kind of treatment is immunotherapy, wherein what is done is through immune molecules, there is desensitization happening. So, what is hypersensitized can be desensitized. Also, what has been done is several exposures of the allergen itself is done in small doses to reduce the IgE on the mast cell and in turn produce more of IgG. So that is something that is there, but most commonly what is done is pharmacotherapy. Okay. Now let us therefore make the conclusions. Type 1 hypersensitivity differs from a normal immune response in the fact that it is mediated through IgE and not the other immunoglobulins. On first exposure to allergen, the system gets sensitized, and on second exposure, there is an acquired response, which is a heightened response. The allergen on binding to IgE on the mast cells or the basophils cross-links the Fc receptors. So what is important is cross-linking the receptors and therefore elicits a signal transduction that leads to degranulation with release of primary and secondary mediators. So type 1 hypersensitivity can be immediate or late, systemic or localized, can be atopic and can be also fatal in many cases. So type 1 hypersensitivity can be an over-the-top immune reaction when exposed to allergens, which can be fatal or merely annoying. Many can be genetically predisposed or may acquire it over a period of time. So wheel and flare is the way of revealing an allergen. So if one wants to do a test, then one should go in for doing a skin test where the symptoms or the telltale signs are wheel and flare. So therefore, in many products that are available in the market, especially cosmetics, you would find that it is written before use, try on in a small part of the skin to see whether you get an allergic reaction or not. That is what is type 1 hypersensitivity. Thank you.